Chapter 5 Molecular Basis of Inheritance. In the previous chapter, you have learned the inheritance patterns and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material at least for the majority of organisms. In class 11, you have learned that nucleic acids are polymers of nucleotides. Deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, are the two types of nucleic acids found in living systems. DNA acts as the genetic material in most of the organisms. RNA, though it also acts as a genetic material in some viruses, mostly functions as a messenger. RNA has additional roles as well. It functions as adapter, structural, and in some cases as a catalytic molecule. In class 11, you have already learned the structures of nucleotides and the way these monomer units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, that is, transcription, the genetic code that determines the sequences of amino acids in proteins, the process of protein synthesis, that is, translation, and the elementary basis of their regulation. The determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is, the DNA. In the subsequent sections, we will understand that why it is the most abundant genetic material and what its relationship is with RNA. 5.1 The DNA. DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides, or a pair of nucleotides referred to as base pairs present in it. This also is the characteristic of an organism. For example, a bacteriophage known as 5x174 has 5386 nucleotides. Bacteriophage lambda has 48502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pairs, and haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. Let us discuss the structure of such a long polymer. 5.1.1 Structure of polynucleotide chain. Let us recapitulate the chemical structure of a polynucleotide chain, that is DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, that is ribose in case of RNA and deoxyribose for DNA, and a phosphate group. There are two types of nitrogenous bases: purines, that is adenine and guanine, and pyrimidines, that is cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. A nitrogenous base is linked to the OH of the first carbon of pentose sugar through an N-glycosidic linkage to form a nucleoside, such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. When a phosphate group is linked to the OH of the fifth carbon of a nucleoside through phosphoester linkage, a corresponding nucleotide or deoxynucleotide, depending upon the type of sugar present, is formed. Two nucleotides are linked through 3 to 5 phosphodiester linkage to form a dinucleotide. more nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free phosphate moiety at 5' end of sugar, which is referred to as 5' end of the nucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH of 3' group, which is referred to as 3' end of the polynucleotide chain. The backbone of a polynucleotide chain is formed due to sugar and phosphates, the nitrogenous bases linked to sugar moiety project from the backbone. In RNA, every nucleotide residue has an additional OH group present at 2' position in the ribose. Also, in RNA, the uracil is found at the place of thymine, 5-methyl-uracil, another chemical name for thymine. DNA as an acidic substance present in nucleus was first identified by Friedrich Miescher in 1869. He named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence in other strand can be predicted. Also, if each strand from a DNA, let us call it as a parental DNA, acts as a template for synthesis of a new strand, the two double-stranded DNA, let us call them as daughter DNA, thus produced, would be identical to the parent DNA molecule. Because of this, the genetic implication of the structure of DNA became very clear. The salient features of the double helix structure of DNA are as follows. 1. It is made of two polynucleotide chains, where the backbone is constituted by sugar phosphate and the bases project inside. 2. The two chains have anti-parallel polarity. It means if one chain has the polarity 5' to 3', the other has 3' to 5'. 3. The bases in two strands are paired through hydrogen bonds, that is H bonds, forming base pairs. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. As a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of the helix, referred to figure 5.2. 4. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometers. A nanometer is 1 billionth of a meter, that is 10 to the power minus 9 meter. And there are roughly 10 base pairs in each turn. Consequently, the distance between a base pair and a helix is approximately 0.34 nanometer. 5. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bonds, confers stability of the helical structure, referred to figure 5.3. Compare the structure of purines and pyrimidines. Can you find out why the distance between two polynucleotide chains in DNA remains almost constant? The proposition of a double helix structure for DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA to protein. In some viruses, the flow of information is in reverse direction, that is from RNA to DNA. Can you suggest a simple name to the process? Central dogma. DNA undergoes replication, then transcription to form mRNA, then translation to form protein. 5.1.2 Packaging of DNA helix. Taken the distance between two consecutive base pairs as 0.34 nanometer, that is 0.34 into 10 to the power minus 9 meters, if the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of pp base pairs with distance between two consecutive base pairs, that is 6.6 into 10 to the power 9 base pairs into 0.34 into 10 to the power minus 9 meters per base pairs, it comes out to be approximately 2.2 meters. A length that is far greater than the dimension of a typical nucleus, that is approximately 10 to the power minus 6 meters. How is such a long polymer packaged in a cell? If the length of E. coli DNA is 1.36 millimeters, can you calculate the number of base pairs in E. coli? In prokaryotes, such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have positive charges in a region termed as nucleotide. The DNA nucleotide is organized in large loops held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquires charge depending upon the abundance of amino acids, residues with charged side chains. Histones are rich in the basic amino acid residues, that is lysine and arginine. Both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome, referred to figure 5.4a. A typical nucleosome contains 200 base pairs of DNA helix. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin, thread-like, stained or colored bodies seen in nucleus. The nucleosomes in chromatin are seen as beads on string structure when viewed under electron microscope EM, referred to figure 5.4b. Theoretically, how many such beads or nucleosomes do you imagine are present in a mammalian cell? The beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as non-histone chromosomal NHC proteins. In a typical nucleus, some region of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are called as heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. 5.2 The search for genetic material. Even though the discovery of nuclein by Michel and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism, that is a bacteria, had changed in physical form. When Streptococcus pneumococcus bacteria are grown on a culture plate, some produce smooth, shiny colonies, S, while others produce rough colonies, R. This is because the S strain bacteria have a mucus or a polysaccharide coat, while R strain does not. Mice infected with the S strain that is virulent die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. S strain inject into mice, mice die. R strain inject into mice, mice live. Griffith was able to kill bacteria by heating them. He observed that heat-killed S strain bacteria injected into mice did not kill them. When he injected a mixture of heat-killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. S strain heat-killed. Inject into mice, mice live. S strain heat-killed plus R strain live, inject into mice, mice die. He concluded that the R strain bacteria had somehow been transformed by the heat-killed S strain bacteria. Some transforming principle transformed from the heat-killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiments. Biochemical characterization of transforming principle. Prior to the work of Oswald Avery, Colin MacLeod, and Macklin McCarty, 1933-44, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals, that is proteins, DNA, RNA, etc. from the heat-killed S cells to see which ones could transform live R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzymes, proteases, and RNA digesting enzymes, RNases, did not affect transformation. So, the transforming system was not a protein or RNA. Digestion with DNAs did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between DNAs and DNAs? DNAS and DNASE? Question mark. 5.2.1. The genetic material is DNA. The unequivocal proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase in the year 1952. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey-Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. They grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA, but not radioactive protein because DNA contains phosphorus, but protein does not. Similarly, viruses grown on radioactive sulfur contained radioactive protein, but not radioactive DNA because DNA does not contain sulfur. Radioactive phages were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from virus to bacteria. Refer to figure 5.5. 5.5 is the Hershey-Chase experiment, where you can see bacteriophage, radioactive S35 labeled protein capsule, and the one is radioactive P32 labeled DNA. First process infection, 2 blending, 3 centrifugation. S35, radioactive S35 detected in supernatant. No radioactivity detected in supernatant in case of P32. No radioactive S35 detected in cells. Please refer to the textbook. 5.2.2 Properties of genetic material, DNA versus RNA. From the foregoing discussion, it is clear that the debate between proteins versus DNA as the genetic material was unequivocally resolved from Hershey-Chase experiment. It became an established fact that it is DNA that acts as genetic material. However, it subsequently became clear that in some viruses, RNA is the genetic material. For example, tobacco mosaic viruses, QB bacteriophage, etc. Answer to some of the questions, such as why DNA is the predominant genetic material, whereas RNA performs dynamic functions as of messenger and adapter, has to be found from the differences between chemical structures of the two nucleic acid molecules. Can you recall the two chemical differences between DNA and RNA? A molecule that can act as a genetic material must be fulfill the following criteria. 1. It should be able to generate its replication. 2. It should be stable chemically and structurally. 3. It should provide the scope for slow changes, that is mutation, that are required for evolution. 4. It should be able to express itself in the form of Mendelian characters. If one examines each requirement one by one, because of rule of base pairing and complementarity, both the nucleic acids DNA and RNA have the ability to direct their duplications. The other molecules in the living systems, such as proteins, fail to fulfill first criteria itself. The genetic material should be stable enough not to change with the different stages of life cycle, age, or with change in physiology of the organism. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that heat which killed the bacteria at least did not destroy some of the properties of the genetic material. This now can easily be explained in light of the DNA and the two strands being complementary if separated by heating come together when appropriate conditions are provided. Further, 2OH group present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. RNA is also known to be a catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Detailed discussion about this requires understanding of the process of repair in DNA, and you will study these processes in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable, mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve fast. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. 5.3 RNA World. From foregoing discussion, an immediate question becomes evident. Which is the first genetic material? It shall be discussed in detail in the chapter on chemical evolution, but briefly we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double-stranded and having complementary strand further resists changes by evolving a process of repair. 5.4 Replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for the genetic material." Watson and Crick, 1953. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. Refer to figure 5.6, which speaks about Watson-Crick model for semi-conservative DNA replication. 5.4.1 The experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in Escherichia coli and subsequently in higher organisms such as plants and human cells. Matthew Mieselson and Franklin Stahl performed the following experiment in 1958. 1. They grew E. coli in a medium containing 15NH4Cl. N15 is the heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA as well as other nitrogen-containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride CsCl density gradient. Please note that N15 is not a radioactive isotope, and it can be separated from N14 only based on densities. 2. Then they transferred the cells into a medium with normal NH414Cl and took samples at various definite time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. The various samples were separated independently on CsCl gradients to measure the densities of DNA. Refer to figure 5.7, which shows Mieselson and Stahl's experiment. Can you recall what centrifugal force is and think why a molecule with higher mass or density would sediment faster? The results are shown in figure 5.7. 3. Thus, the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid or intermediate density. DNA extracted from the culture after another generation, that is after 40 minutes, that is the second generation, was composed of equal amounts of this hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportions of light and hybrid densities DNA molecule? Very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes was performed on Vicia faba beans by Taylor and colleagues in 1958. The experiments proved that DNA in chromosomes also replicates semi-conservatively. 5.4.2 The machinery and the enzymes. In living cells, such as E. coli, the process of replication requires a set of catalysts, that is, enzymes. The main enzyme is referred to as DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli, that has only 4.6 into 10 to the power 6 base pairs, compare it with human whose diploid content is 6.6 into 10 to the power 9 base pairs, completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pairs per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during replication would result into mutations. Furthermore, energetically, replication is a very expensive process. Deoxyribonucleoside triphosphates serve dual purposes. In addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphates in a dinucleoside triphosphates are high energy phosphates, same as in case of ATP. In addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occurs within a small opening of the DNA helix referred to as the replication fork. The DNA-dependent DNA polymerases catalyze polymerization only in one direction, that is from 5' to 3'. This creates some additional complications at the replicating fork. Consequently, only on one strand, the template with polarity 3' to 5', the replication is continuous, while on the other, the template with polarity 5' to 3', it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase, referred to figure 5.8. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite region in E. coli DNA where the replication originates. Such regions are termed as origin of replication. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during the recombinant DNA procedures, requires a vector. The vectors provide the origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results into polyploidy, that is a chromosomal anomaly. You will learn the detailed nature of origin and processes occurring at this site in higher classes. 5.5 Transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Here also, the principle of complementarity governs the process of transcription. Except the adenosine complements now forms base pair with uracil instead of thymine. However, unlike in the process of replication, which once set in, the total DNA of an organism gets duplicated, in transcription, only a segment of DNA and only one of the strands is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Why both the strands are not copied during the transcription has the simple answer. First, if both strands act as a template, they would code for RNA molecule with different sequences. Remember, complementarity does not mean identical, and in turn, if they code for proteins, the sequence of amino acids and in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a futile one. 5.5.1 Transcription Unit. A transcription unit in DNA is defined primarily by three regions in the DNA. 1. A promoter. 2. The structural gene. 3. A terminator. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA-dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5' to 3', the strand that has polarity 3' to 5' acts as a template and is also referred to as template strand. The other strand, which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil, is displaced during transcription. Strangely, this strand, which does not code for anything, is referred to as coding strand. All the reference point while defining a transcription unit is made with coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. 3' ATGC 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 5', that is template strand. 5' TACG 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 3', that is coding strand. Can you now write the sequence of RNA transcribed from the above DNA? Figure 5.9 Schematic structure of a transcription unit. The promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards 5' end upstream of the structural gene. The reference is made with reference to the polarity of coding strand. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in the transcription unit that also defines the template and coding strands. By switching its position with the terminator, the definition of coding and template strands could be reversed. The terminator is located towards 3' end downstream of the coding strand, and it usually defines the end of the process of transcription. Refer to figure 5.9. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with the regulation of gene expression. 5.5.2 Transcription unit and the gene. A gene is defined as the functional unit of inheritance. Though there is no ambiguity that the genes are located on the DNA, it is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for tRNA or rRNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said as a monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. In eukaryotes, the monocistronic structural gene have interrupted coding sequences. The genes in eukaryotes are split. The coding genes or expressed sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed DNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as a regulatory genes, even though these sequences do not code for any RNA or protein. 5.5.3 Types of RNA and their process of transcription. In bacteria, there are three major types of RNAs: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template, tRNA brings amino acids and reads the genetic code, and rRNA plays structural and catalytic role during translation. There is a single DNA-dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription, that is, initiation. It uses nucleoside triphosphates as substrates and polymerizes in a template-dependent fashion, following the rule of complementarity. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerases reach the terminator region, the nascent RNA falls off. So also the RNA polymerase. This results in termination of transcription. An intriguing question is that how is the RNA polymerase able to catalyze all the three steps, which are initiation, elongation, and termination? The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with the initiation factor, sigma, and termination factor, rho, to initiate and terminate the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. Refer to figure 5.10. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation take place in the same compartment, there is no separation of cystole and nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. In eukaryotes, there are two additional complexities. One, there are at least 3 RNA polymerases in the nucleus. That is, in addition to the RNA polymerase found in the organelles. There is a clear-cut division of labor. The RNA polymerase 1 transcribes rRNAs, that is 28S, 18S, and 5.8S, whereas the RNA polymerase 3 is responsible for transcription of tRNA, 5SR RNA, and snRNAs, that is small nuclear RNAs. The RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA, hnRNA. 2. The second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. Hence, it is subjected to a process called splicing, where the introns are removed and exons are joined in a defined order. hnRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide, that is methyl guanosine triphosphate, is added to the 5' end of hnRNA. In tailing, adenylate residues, about 200 to 300, are added at 3' end in a template-independent manner. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation. Refer to figure 5.11. The significance of such complexities is now beginning to be understood. The split gene arrangements represent probably an ancient feature of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In recent times, the understanding of RNA and RNA-dependent processes in the living system have assumed more importance. 5.6 Genetic code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There existed ample evidences, though, to support the notion that change in nucleic acids, genetic material, were responsible for change in amino acids and proteins. This led to the proposition of a genetic code that could direct the sequence of amino acids during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and deciphering of genetic code were most challenging. In a very true sense, it required involvement of scientists from several disciplines: physicists, organic chemists, biochemists, and geneticists. It was George Gamow, a physicist, who argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because a permutation combination of 4 cube 4 into 4 into 4 would generate 64 codons, generating many more codons than required. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khorana was instrumental in synthesizing RNA molecules with defined combinations of bases, homopolymers and copolymerase. Marshall Nierenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severo-Ochoa enzyme, that is polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequences in a template-independent manner, that is enzymatic synthesis of RNA. Finally, a checkerboard for genetic code was prepared, which is given in table 5.1. 5.1. The codons for various amino acids: UUU phenylalanine, UUC phenylalanine, UUA, UUG, CUU, CUC, CUA, CUG, leucine, AUU, AUC, AUA, isoleucine, AUG, methionine, GUU, GUC, GUA, GUG, valine, UCU, UCC, UCA, UCG, serine, CCU, CCC, CCA, CCG, proline, ACU, ACC, ACA, ACG, threonine, GCU, GCC, GCA, GCG, alanine, UAU, UAC, tyrosine, UAA, UAG, stop codon, CAU, CAC, histidine, CAA, CAG, glycine, AAU, AAC, asparagine, AAA, AAG, lysine, GAU, GAC, aspartic acid, GAA, GAG, glutamic acid, UGU, UGC, cysteine, UGA, stop codon, UGG, tryptophan, CGU, CGC, CGA, CGG, arginine, AGU, AGC, serine, AGA, AGG, arginine, GGU, GGC, GGA, GGG, glycine. The salient features of genetic code are as follows. 1. The codon is triplet. 61 codons for amino acids and 3 codons do not code for any amino acids, hence they function as stop codons. 2. Some amino acids are coded by more than one codon, hence the code is degenerate. 3. The codon is read in mRNA in a contiguous fashion. There are no punctuations. 4. The code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine, PHE. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. 5. AUG has dual functions. It codes for methionine, MET, and it also acts as initiator codon. 6. UAA, UAG, UGA are stop terminator codons. If following is the sequence of nucleotides in mRNA, predict the sequence of amino acid coded by it. Take help of the checkerboards. AUG, UUU, UUC, UUC, UUU, UUU, UUC. Now try the opposite. Following is the sequence of amino acids coded by an mRNA. Predict the nucleotide sequence in the RNA. Methionine, phenylalanine, 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 phenylalanine. Do you face any difficulty in predicting the opposite? Can you now correlate which two properties of genetic code you have learned? 5.6.1 Mutations and genetic code. The relationships between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in Chapter 4. Effects of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutations will be explained here. A classic example of point mutation is a change of single base pair in the gene for beta-globin chain that results in the chain of amino acid residue glutamate to valine. Point mutation. It results into a diseased condition called as sickle cell anemia. Effect of point mutations that inserts or deletes a base in a structural gene can be better understood by following simple example. Consider a statement that is made up of the following words, each having 3 letters like genetic code. Ram has red cap. If we insert a letter B in between has and red and rearrange the statement, it would read as follows. Ram has bre DCAP. Similarly, if we now insert 2 letters at the same place, say BI, now it would read Ram has ber EDCAP. Now we insert 3 letters together, say big. The statement would read Ram has big red cap. The same exercise can be repeated by deleting the letters RED one by one and rearranging the statement to make a triplet word. Ram has EDCAP, Ram has DCAP, Ram has cap. The conclusion from the above exercise is very obvious. Insertion or deletion of one or two bases changes the reading frame from one point of the insertion or deletion. However, such mutations are referred to as frame shift, insertion or deletion mutations. Insertion or deletions of 3 or its multiple bases inserts or deletes in one of the multiple codon, hence one or multiple amino acids, and reading frame remains unaltered from that point onwards.